Good morning, everyone, and welcome to another CALIN webinar in our 2020 series. This is the final webinar for Series 1. We're very lucky today to have an additional talk provided by Dr. Lewis Francis. So Dr. Francis is he's an associate professor at Swansea University in the medical school. He's part of the reproductive biology and gynaecological oncology research group. And he also, for CALIN, he's one of our CALIN academics on the project, and he's also the medical school director for another collaborative project called the Avenues of Commercialization for Nano and Microtechnologies, the ACNM project. So Lewis's background is firmly rooted in biophysics and epigenetics, and so he's going to give us a webinar today focusing on the topic listed there: epigenomics and disease management, functional collaboration towards a high content future. There will be an opportunity for some brief questions at the end. So if you do have any questions, please use the webinar control panel to either type them into the chat window or the questions section, and we'll feed those back to Lewis at the end of the presentation. So I'll hand over to Lewis, and he can take you through the webinar this morning.
Good morning, everyone. My name is Lewis Francis. I'm an associate professor in the Reproductive Biology and Gynaecology group in Swansea University. And I talked to you this morning about our role in CALIN and how we've managed to use the network to really develop our results and our research and our hypothesis-led research around epigenomics, disease management, trying to set the foundation really for future high content analysis to try and stratify disease and develop advanced therapeutics. So what is meant by the term epigenomics really is the study of understanding kind of nuclear architecture from DNA-protein interactions in the form of chromatin, and how chromatin is changed, or how chromatin is regulated, and the structure and the function of chromatin from silent genes, where the chromatin, the DNA-protein complexes are compressed and there's no room for transcription, so usually it's a repressive state and the genes are switched off. To then active or euchromatin states, where DNA-protein complexes are loosely attached, you have open reading frames and you have active transcription. And in between those, you get these poised or bivalent chromatin states, which give plasticity, give the opportunity for cells to react quickly in their environment, and they can react to biochemical cues, biomechanical cues, and they can really connect genes and transcriptome with the local environment of the cell. So we really developed this highly evolved, highly complex series of post-translational modification mechanisms that act on histone complexes, so histone modification, DNA methylation, non-coding RNAs. So they're the main mechanisms of epigenetic regulation that really play this complex, interrelated relationship with the transcriptome, with the active gene expression, which controls cell function and cell fate. And epigenomics is exciting. It's really taken off in the last 20, 30 years. And they've been linked, with profiles to function in epigenomics, from pharmacoepigenomics, so that these post-translational modifications are reversible, so we can target them in terms of advanced therapeutics and translate them to drug development and pharmaceutical industry. Technologies and biotechnology have really seen a boom in epigenetic and epigenomic technologies and applications in the last 10 years, where we can now go down to single cell resolution and understand cell heterogeneity. And that single cell epigenomics kind of allows us to look into the past of the cell. Where's that cell come from? How's that cell been involved in development, disease, tumorigenesis, for example, and really predict the future fate of that cell based on its chromatin architecture and its gene expression profiling. And we do that at a global scale, so we're talking genomic wide. So if you think of it as a linear piece of beads on a string, most people will have seen the beads on a string kind of cartoon that we see here. We know that genes have promoters, they have regulatory regions where transcription factors and other complexes form in proximal promoters. They form transcription initiation complex, elongation complexes that then transcribe the gene. We know that there are upstream regulatory elements such as enhancers that have huge effects on where the genes are expressed. And because of the 3D architecture, the kind of boulder spaghetti cartoon of chromatin and nuclear architecture, we know that distal sites way upstream can have effect in gene transcription. So transcriptional and epigenomic machinery, they work in concert, they work together, it's coordinated, it's transient, it's dynamic. And this transcriptional cooperation and the identification of the regulatory hierarchies really have demonstrated the importance of epigenetic regulators in mitogenic control and potential as therapeutic targets. So when I talk about high content analysis, I'm talking about complex kind of multi-platform analysis that reveals this landscape, this molecular landscape in a single cell between epigenomic and transcriptomic cooperation. And we're particularly interested in looking at cancer and using that to understand functional avenues that we can really take advantage of as we drive towards personalized medicine. Okay, so how do we go around? What's our approach in Swansea? So we have an interdisciplinary approach, and if you think of it as academics for business within the umbrella of the Callan network, we're really using academic innovation and business industry motivation to try and drive sector development. So we have collaborative innovation around cancer epigenomics in my particular interest. So it started off as academic interest, what drives transcription, what controls individual genes, particularly in my case, genes that code for proteins that are functional at the cell surface and drive cell-cell interaction. Clinical need, so understanding the clinical need. So, in cancer epigenomics, how can we use it? How can we answer and develop methodologies that will help clinicians find the answers that they need to help patients in a better way? And biotechnology, really. So how do we develop the tools to understand? So I'll use you an example of the previous version of Callan with CAN, and now leading into Callan, where we've developed biotechnologies and antibody-based assays to try and understand the role of different proteins in these chromatin, DNA protein complexes. And I'll talk a little bit more about that today. So really the take-home message for us is that you have to have multidisciplinary teams, industry academic teams, and you have to have common ground and collaborative questions with everyone benefiting in order to really make great strides towards high content analysis. This is a great example of that. So we have Reproductive, which is the life science brand of Biomedical Sciences, which is based up in North Wales. And they've really benefited, and we really benefited from working alongside them to take the inert porous plastic filters, use different chemical functionalizations, and understand how we can use it like CHIP seq. So CHIP is chromatin immunoprecipitation, where we use different antibody-based assays to isolate different regulatory proteins and their location within the DNA genome. So by using the antibody, you can pull down the protein, and then by pulling down the protein, you can separate the DNA and use PCR or next-generation sequencing libraries to understand where that protein is located. And then by doing some nice informatics and some high-content analysis, you can understand if that protein was located in gene proximal promoters or in other genome landmark sequences, such as those upstream distal enhancers. And you start to really get these molecular networks of regulation. And if you do RNA transcriptome next to this, then you can get that balance between epigenetic and transcriptome analysis. So we go back a long way with Chromatrap, and we've developed and been involved in the development of the kit and the assay, which is now successfully launched to market. And the Catholic Alliance funding has been integral process to that. So we now have an ability, we have a suite of assays, a catalogue of assays, to look at different transcription factor marks listed on the left-hand side. So we can really start to unravel this cartoon and unravel the landscape and understand what's going on. So I'm going to talk to you a little bit more about how we've developed that now and some applications around the science. I'm not going to go into the science, I'm just going to go into the overall approach. and understand what we're trying to do. And what we're really trying to do is develop omics approaches. So downstream of those interesting assays that we've developed, we've now developed next generation sequencing. So we can go transcriptome, RNA-seq, we can go single-cell ATAC-seq, which is a way of understanding if the culminating architecture is open or closed through nucleosome-free regions. And then once we understand if there's a change, a gross change in chromatin architecture, we can understand ChIP-seq, which allows us to look at the different histone marks, for example, or transcription factors, and understand how they are managing that chromatin accessibility in open and closed landscapes. And then we can also look at DNA and methylation-seq, where we look to see if the DNA methylation landscape is involved. So we can start to develop this multi-parameter, multi-omics approaches to understand different cells in a cancer landscape, for example, different cells in a catalogue of immune cells, and really start to decipher the molecular heterogeneity seen. And you can go all the way down to single cell resolution now. So the high content analysis comes from very large data, very intricate IT networks, very intricate mapping of these molecular networks to try and understand what are the regulatory pathways, what is the interaction between transcription factor networks, chromatin signatures, and then the genes that are transcribed that drive cell fate. And what I don't have on this slide is kind of cell imaging, so we can do exactly this with an image of a cell with multiple different fluorescent probes, and we can understand whether these pathways are directly related and are driving a particular disease phenotype. So once we pull all this together, we have multiple applications and multiple data sets that we then can do multivariate statistics on, and we can start to drive towards high content analysis, deconvolution of tumor heterogeneity, understanding if there are single elements of cells or populations of cells within a different organ system during disease or during development that are the drivers of that phenotype presentation. And then whether or not different drugs have an effect, whether or not the different subtypes of disease change, what are the role of different proteins within those diseases when we understand expression networks. And that's what we're trying to do in RBGO, in our team, is try to provide these multi-omics approaches for clinicians, for industry partners, and for ourselves to really drive new knowledge, particularly in ovarian cancer in our space. We do that through building workflow-based solutions, so it's easy to have the dream of how to do it, but how do you do it? So we understand the unmet clinical needs, starting at the top left of the slide. And then we roll through in vitro models. So we develop in vitro models, both in 2D and 3D, with co-cultures, single cultures. And we understand how cells come together and how they interact in that tissue space. And then we've developed the genomic material isolation techniques, and the assays, as I mentioned, with Chromatrap as one example. Library preparation for next generation, all of the quality controls and understanding that we're putting good quality nuclear or genomic material into the assay and into the sequencing-based assays. We've developed sequencing relationships with industry companies in Europe, worldwide, through the US, but also in Wales. So Wales now, with the Wales Genome Park, are investing heavily in next-generation sequencing and trying to understand this clinical and fundamental research interface and how we can come together and really drive analytical platforms so that we can develop opportunities through understanding single-cell heterogeneity, deconvoluting tumor microenvironment, and driving towards personalized medicine. And my talk, basically, I have some very basic research slides now showing examples of that from an exciting epigenomics advanced therapeutics project we have with a cluster of epigenomics and advanced therapeutics. A Callan studentship where Francesca, a student of ours, is working with a jellyfish company looking at cells and how they interact with collagen so that we can understand how matrix drive, extracellular matrix can drive tumorigenesis in ovarian cancer. Cancer survivor study, where we've been really excited to be involved in an amazing project that's really linking these cancer survivors and patient need with some of our high throughput approaches, and also with therapeutic exosomes, where we're understanding how we can treat and develop these therapeutics. So ovarian cancer, very, very aggressive, very metastatic. It's got the highest mortality rate of any gynecological cancer. It's a very heterogeneous disease. It's almost classed as a subset of different diseases. It derives from the ovarian epithelial cells. And after initial tumorigenesis, it progresses very quickly to the metastatic state where the cells leave primary tumor and metastasize to the peritoneal space. It's high-grade serous, and it's got multiple different subtypes. So interestingly, ovarian cancer has got a very low mutational profile, so a very simple mutational profile where it's got some mutations in genes. But the genomic mutations are simple, low, and it's kind of known unofficially as the epigenetic disease. So high-grade serous, most common, very aggressive, and extensive structural genomic variations can alter this epigenetic landscape. And we've put together a cluster of interested industrial partners to really try and pull apart the epigenomic landscape of ovarian cancer, both from stratifying patients to then developing drugs that can target the epigenetic landscape. How are we going around doing that? So we can do it both from taking patient samples, producing large omics data approaches from different subtypes and looking at the epigenetic trends. We can take data that's already published, data that's publicly available, and look for trends. So these are particularly the threads through the slides here, the enhancer sites. So what are the predicted enhancers? So we can then say, okay, what are these footprints for epigenomics in terms of histone profile distribution, recurrence rates, genomic locations, association between different gene families and profiles, overlap with previously identified enhancers, and there's a lot of interesting data coming out around enhancer activity being linked to cancer resistance, for example, and then linking that to pathway and doing some go analysis to look to see if we can understand the functional consequence of these epigenomic patterns. And this is really underway now at the moment. And then, as well as taking the subtypes, characterizing the epigenetic footprints, we can also then take drugs, for example, and we can understand whether or not different drugs, in our case, epigenetic compounds provided by GSK, can affect the epigenetic landscape, can reverse cellular phenotype and drive towards tailored therapy. By understanding the signatures in our patient stratification, we can then tailor therapies once we understand the mechanism of action of drugs to really analyze large data sets to drive functional research questions and stratify patient groups and predict cell fate. And then along those lines, really, ovarian cancer doesn't happen on its own. It's a heterogeneous mix of different cell types. Those cell types exist within a matrix environment. They have a structure. They interact with extracellular matrix. They secrete extracellular matrix, and they need to break free from that environment to drive metastatic spread, get out into the vasculature, and become this metastatic aggressive disease. So one of my personal interests is understanding, are the cells driving the extracellular matrix, or is the matrix driving the cells? And we know that, patient biopsies become stiffer, for example, when disease is at a more advanced stage. So Francesca's working in a Cali-funded PhD. She's just coming to the end of her second year now. It's not actually 2.5 years in, and she's understanding how to use different types of collagen to model the matrix, to see how the matrix and the mechanical pressure in a tumor environment can induce epigenetic changes. So she's taken a jellyfish collagen with gelatin. She's understood that cancer cells are compatible with that structure. They can dive down into that structure and make a 3D tumor. And now we're modifying the basic collagen structures through some post-translational modifications and some activation to understand if we can drive different chromatin states and different levels of tumorigenicity based on the stiffness of the matrix around the cells. And we can see that as we change the stiffness, we can change the activation state of ovarian cancer cells, and we're now characterizing and isolating the chromatin to understand the landscape in those cell types. Another example of an exciting project is understanding if we can model human peripheral blood cells, so PBMCs. So we take a mixed population, isolate, genomic material, and isolate the chromatin, isolate DNA to understand the methylation profiles, and really see if we can understand from the patterns of the chromatin and the omics taken from those mixed PBMC types, whether or not we can use genomics, transcriptomics, epigenomics to really characterize the different types of cells in different patient groups. So for example, in our study, it's looking at patient groups that can survive cancer. Can we phenotype stratify those patients against patients who unfortunately can't survive cancer or are cancer sufferers, and then comparing those back to the healthy population to understand how epigenomic plasticity in PBMCs is related to our ability to fight off or deal with suffering from this terrible disease. So I'm just coming to the end now, but what I wanted to capture today is that we work with industry partners, and we're very fortunate, all of us, to be part of this CALIN network that gives us funding to work with industry partners, work with other academic partners, like-minded people who have, such as Frank in Galway, who have great ideas about how we can deconvolute genomic patterns of gene expression, transcriptomics, and epigenomics to try and understand these regulatory footprints. And we need to do that from biotechnology, so understanding that we have the right tools, to the right cellular models, and we know that we're talking to the clinicians and we are answering questions that they need answered. So we're really driving towards industry development by providing the tools needed by the industry to answer interesting questions, and that in turn drives sector development through drug development or stratified medicine in order to give clinicians the answer they need for next generation clinical management. So where transcriptomics epigenetic profiling to understand regulatory footprints, how those regulatory footprints can drive mitogenic or metabolic gene networks, so that we can understand the role that different transcription factors, different complexes and interplay in epigenetics, what the role is in tumorigenesis, metastatic development, and also how we can reverse those traits to try and provide opportunities for better clinical management. These industrial, academic, clinical collaborative networks, they lead to enhanced understanding of global patterns. They lead to our ability to think outside the box, to ask big questions, and to become high content, so that we can drive and convert specific transcription responses and understand cell failure programs in our different disease areas. And all of the approaches I'm talking about today are translatable to different disease areas. What are the opportunities? So hopefully I managed to convey to you today just how high content this is. So by taking regulatory co-activation networks, we can look at epigenomics, transcriptomics, we can do genomic mapping, transcriptome mapping, by understanding the peaks, so where different proteins and different histone marks sit, for example. We can understand regions that we should be looking at, what the genes are in those regions, and where and what proteins and what modifications are happening, so that we can really understand these molecular maps and cooperation networks between epigenetic master regulators, families of genes, upstream regulatory elements, for example, that may be having an effect. And as we build these high content data, so all genome based, large data sets, as we're building these profiling data sets, we can then start to teach machines to understand them, to look at networks, to look at how networks are modified in the case of different patient groups, modified in the case of different drugs. So then we can start putting those together, and we can say, okay, tell me what drug would be better tailored for different patient groups. And in Swansea, we're well on the way to doing that. We can't do that without industrial collaboration and without forward-thinking funding networks such as CALIN. And we're therefore very, very grateful to be able to tell you guys about this today and explore this as part of the webinar and think about future collaborations and how we can really deliver to improve clinical management strategies. So in summary then, we have industry academic collaborations across the world We have academic collaborations, for example, in Grenoble, in Europe, in Ireland, in the States, in University of Houston, and in Methodist. In the Methodist, we're building on Calin collaboration to really take Welsh-Irish interaction and drive it with US partners to really improve and increase high impact factors. And that's led us to develop tools that has allowed us to work with large pharma, with other industry partners that are anchor companies for us in our group now in RBGO, publish high impact factor papers, and really think big in terms of where we can go in terms of the future and get excited. Not just from fundamental research, but in terms of growing life science industry, in terms of tailoring our therapies for bespoke clinical needs in our part of the world, and also really to deliver from a Pan-Wales point of view to help develop next generation analysis support and interpretation at the clinical interface through Wales Genome Park, and always trying to think about the needs of local NHS and international healthcare services to try and make a difference ultimately. So that's me done for today. Thank you for listening. We have a talented team to RBGO. We have excellent funding. I'm very grateful to be part of the network and to lead the network and work with everyone and work with like-minded people. Thank you to our patients for providing samples. Thank you to all the guys in the team who are, they are a talented team and excellent. Everyone works together. Everyone's motivated and enthusiastic. And we can't collaborate and play active roles and collaborations without the team on the ground. So thanks. And yeah, be happy to take questions.
Brilliant. Thank you for that excellent talk, Lewis, giving us a real insight into the research that you guys are conducting from RBGO, but also from the wider perspective and all these different collaborative projects which enable to really foster innovation moving forward. And it's really interesting to see how these are moving forward on an international basis with the links into the US and places like Grenoble. So it's fascinating to see this breadth of collaborative research that's being done out of Swansea and being led by the guys from the medical school in Swansea. So it's really, really fascinating. So I want to thank you for your time and effort for giving us that brilliant webinar. Before we move on to questions, again, as I said at the beginning, if anyone does have questions, please put them in the question or chat window, and I'll put those to Lewis in just a second. I just wanted to highlight, I said at the very beginning that this is the last talk in the first series of webinars for the Calin Series 2020. So there's going to be a couple of week break now until we have our Series 2 webinars. Now, our Series 2 webinars have a bit of a different theme to them. The ones we've had so far is sort of giving an insight into the teams and groups behind the research. And the next set of webinars are looking at some of the projects we've actually conducted within Calin and how the companies have found that process and how it's benefited them. So it's called How Has Calin Helped Process and Benefits and Insight, and it begins on the 28th of July and it runs for 6 weeks then right through to the 1st of September. So the next one up in that series and the series overall will be Calin case studies from University College Dublin, UCD. So they're going to take us through a couple of the projects that they have been working on, and we're going to get a company perspective of how they found the process and the work that's come out of collaborating. And then this is followed then by the one from Cardiff University on the 4th of August. For more details and registration, please do visit the website, and there's a few more details and the registration form is available. So I'm going to open it out to questions now for Lewis. So if anyone does have any questions, if you do put them in the question or chat window, and I shall just have a look, see what we've got in, and we'll just get Lewis to give us an answer if he can on them. So one of the questions we have here is, what advice would you have, Lewis? We talk a lot about collaborative research and in industry and clinical collaboration. You highlighted that very well on one of your slides in the webinar. What advice would you have if there is a company or a clinician listening in today and is you've sparked an interest in them, and they think, hang on, I might have some ideas around research to be involved here, or I'd like to get involved. How do they go about that? What would be your advice to them?
I think the first thing to do really is to understand, who is in the institute that they particularly paid with, who works in that area, and to reach out and to have a conversation with a like-minded person who may have a similar idea, and to really explore opportunities in-house for internal collaboration. But then hopefully that will then lead them to the Calin network and reaching out to us as a potential partner, or at least a soundboard for those ideas, and see whether those ideas are, or to get different perspective on those ideas. And once you've done that, then you can really, go into the literature, dig and see what other people are doing, both nationally, internationally, and see the trends that talk to that idea. And I think that it can become very clear very quickly whether or not that idea has got some legs, has got future, and people will get excited quite quickly especially if themes around epigenomics, drug development, these multidisciplinary kind of approaches, they can snowball quite quickly. So my advice would always to be clear on kind of what the idea is, and to harness enthusiasm by looking in-house, but also reaching out through the network. And we'd be, for sure, we'd be happy to help to bounce ideas, and not worry about people becoming partners and necessarily sharing IP or anything like that, but just let the idea and the enthusiasm behind the idea take over in the first instance, so that you get the best chance to partner with the right people.
Brilliant, excellent. That's a really great answer. For anyone who's interested, I have put the contact details on the screen there. So I put Lewis's email address up there, l.francis@swansea.ac.uk. But also you can get in touch with Calin in general. Like has been pointed out many times, we've got 6 different university partners, and we will always signpost people to the best area for support, whether it's within Calin or not. So we have a website, we have a Twitter and a LinkedIn page. So please do get in contact with us. As Lewis said, chatting about it and pushing that enthusiasm behind an idea is the first port of call, and everything else can follow on from there. There has another question come in that says, will there be an opportunity for Calin partners to link in with some of the bigger industry partners you mentioned, such as GSK?
Well, for me, it's a really nice question, and one of the really kind of huge advantages of the network is the communication and dissemination of information between partners, highlighting, from case studies through to who the potential partners are, but also linking them to wider kind of engagement opportunities through the university and higher education systems involved. So, as for example, by me highlighting opportunities in epigenetics through GSK, potential partnerships through Swansea, if that can create a communication bridge into GSK for somebody, then absolutely it's a positive. We have good established links with their R&D departments, particularly around oncology, but also access into GSK, which is a big animal. So yes, absolutely is the answer to that question, and we'd be happy to kind of mediate interactions and make introductions for anyone from within the network out to our partners, not just GSK, but all other partners.
Brilliant. Again, another fantastic answer there. I think you've definitely covered the point. We do it within part of Calin as well. I think you mentioned it. We have some strategic partners in Calin, which is GE Healthcare and Unilever. So again, 2 very large companies that we have links and access into. Now, just finally, there is one more question that's come through. It's a bit more of a perspective question rather than a fact-based question, I guess. And it says, given the current climate with COVID and the pandemic, do you foresee that collaborative projects such as Calin and SEAT and the rest of them actually hold even more importance to being able to support companies and industry who may well not be able to pursue their own research and development as easily, as their focus is shift on, maybe potentially just trying to survive and keep the business afloat?
Yeah, I think it's I think it's clear and obvious now that businesses worldwide are being hit. The bottom line of businesses being hit by COVID and some of the restrictions around not only working practices but also international markets. And I think that is going to put an onus on R&D departments to think outside the box, to reach out to partners. It's going to strengthen already existing kind of collaborative relationships, but it's also going to open up doors to new relationships. And by communicating through networks such as this and allowing industry partners to see what's available, by continually kind of being proactive in terms of funding models and how we can best suit to the R&D bottom line in large institutes. I think as long as we can, so yes, the answer I think is going to mean that companies need to reach out and need to have more bespoke R&D portfolios, and that higher education partners and smaller industries and partnering can provide innovation for sure. But it is also an onus on us, and also on us to talk to funding mechanisms, funding bodies, to make sure that we're proactive, that the paperwork trails, etc., and the logistics of working with us as partners are as free and as easy as they can be. I think one of the major bridges is going to be, well, if they see a potential interaction and innovative collaboration and they get excited, but then there are 10,000 hurdles in between the starting point of that interaction. I don't think they're going to have time to really go through very intricate logistical kind of paperwork trails. So we need to streamline those and make it as easy as possible for these companies to come on board.
Brilliant. Again, a brilliant answer there, Lewis. So I don't think there's any more questions that have come in now. So I would just like to thank you for taking the time today to put that webinar together and give us that really, really interesting insight into the world of epigenomics and the collaborative nature of it, and how it's really driving the next stage of research forward. And I'd like to thank all of our listeners for once again attending one of our webinars, and hopefully it's been very informative and beneficial for you. And I'd just like to say, hopefully you can join us for the next series that starts on the 28th of July. And please do keep well and keep safe. Thank you all.